using CRISPR to cure diseases and extend human lifespan. CRISPR-Cas9 was first used as a gene editing tool in 2012. In just a few years, the technology has exploded in popularity, thanks to its pledge of making gene editing briskly, cheaper, and easier than ever before. We are glad you are here. Are you set to catch up on the most incredible future applications of CRISPR gene editing technology? We will examine how CRISPR might influence the future. In the previous video, you have seen how CRISPR could bring extinct species back to life. In this video, we will show you using CRISPR to cure diseases and extend human lifespan, and the ethical considerations of gene modification. Diseases similar as cancer, Alzheimer's disease, HIV, muscular dystrophy, and inherited eye disorders affect millions of people each year but have no cures. Now, CRISPR-grounded therapies for many diseases have formally shown to be effective through mortal trials, steering in a new era of transformative medicine. CRISPR-Cas9 is a gene editing technology that's poised to revolutionize medicine. Researchers are developing CRISPR-Cas9 therapies for a wide range of diseases, including inherited eye diseases, neurodegenerative conditions similar as Alzheimer's and Huntington's diseases, and non-inherited diseases similar as cancer and HIV. In fact, CRISPR mortal trials are already underway for numerous of these diseases. So how can CRISPR be used to cure diseases? Using the CRISPR system, researchers can precisely edit any target DNA locus, a feat that wasn't attainable using other gene editing tools. The possibility to edit a disease mutation to correct inheritable errors creates opportunities for treating conditions that have long escaped the medical research community. What conditions can CRISPR cure? CRISPR can cure a number of inheritable conditions, ranging from blood conditions like sickle cell anemia to cancer. The list of conditions presently being combated by CRISPR has been growing every day. The data from clinical trials released lately has demonstrated that CRISPR remedy has been successful in treating cases with sickle cell anemia as well as beta thalassemia. With the recent development of high editing, which allows targeted small insertions, elisions, and base switching in a precise way, scientists are agitated to add a new tool to the genome editing toolbox, steering in the pledge of fresh treatment options. So let's take a look at how experimenters are using CRISPR to treat these conditions. Using CRISPR for cancer treatment. With its rearmost success in clinical trials for the treatment of several conditions, people are agitated further than ever about this technology and are inclusively pondering the question, can CRISPR cure cancer? A lot of exploration is being done on answering this question, particularly in CAR-T. Let's take a look at some of the cancer scientists are hoping to cure with this technology and the progress they've made so far in both preclinical studies as well as human trials. Lung and esophageal cancers. It is difficult to treat esophageal cancer and some types of lung cancer. Chemotherapy is particularly ineffective against a number of lung cancers, and esophageal cancer is frequently discovered late in its progression, resulting in poor outcomes. According to estimates, 235,760 new cases of lung and bronchial cancer and 19,260 new cases of esophageal cancer are anticipated to be diagnosed in 2021. A protein PD-1 on the surface of cells that downregulates the body's natural immune response has been directly linked to tumor growth in numerous cancers, including lung and esophageal cancers. The self-tolerance of cancer cells is facilitated by PD-1. To enhance T-cell anti-tumor activity, Junhui Kai at Hebei Medical University developed a CRISPR-Cas9-based strategy in which PD-1 is silenced in cancer patient cells. CRISPR-edited T-cells can be used safely and effectively in clinical settings, as demonstrated by the results of a recent clinical trial that looked into CRISPR-Cas9 gene therapy in patients with advanced non-small cell lung cancer. Lung cancer patients' blood was taken out, immune cells containing PD-1 were destroyed, and the blood samples were then reinfused back into the body. Esophageal cancer can be effectively treated with this CRISPR-Cas9 PD-1 knockout therapy. Another clinical trial investigated a similar PD-1 knockout therapeutic approach for treating esophageal cancer. Multiple myeloma. A cancer of the bone marrow known as multiple myeloma is characterized by abnormalities in plasma cells, a type of white blood cell. The abnormalities cause the plasma cells to multiply out of control, becoming the majority of the bone marrow cells and increasing from less than 1% to the majority, resulting in bone tumors. In 2021, 34,920 new cases of multiple myeloma are anticipated to be diagnosed in the United States. CRISPR Therapeutics is investigating the safety and efficacy of a CRISPR-based therapy, CTX120, in subjects with relapsed or refractory multiple myeloma. The dose escalation and cohort expansion study is predicted to be completed in early 2027. Another trial by Allergen is investigating the efficacy of ALO-605, a TALEN-engineered turboCAR treatment currently in preclinical stages. Curing blood diseases using CRISPR technology. Sickle cell anemia and beta thalassemia are life-threatening blood diseases that can only be treated with bone marrow transplants from relatives. Patients, on the other hand, can finally relax knowing that there is finally hope for a cure thanks to the recent success of CRISPR technology. Sickle cell disease is caused by a variation in a single base in the beta-globin gene, one of the genes encoding hemoglobin. It is characterized by red blood cells with abnormal shapes, and its symptoms include severe pain attacks, anemia, and swelling of the limbs, stroke, and bacterial infections. Millions of people worldwide are affected by sickle cell disease. Blood transfusions and bone marrow transplants are the best treatments for the disease, which does not have a cure at this time. CRISPR-based gene therapy has recently demonstrated significant promise as a treatment for this condition. The current treatment options simply address symptoms of sickle cell disease, but CRISPR-Cas9 has demonstrated the potential to cure the underlying genetic cause of the disease. A significant step was made toward this goal when Vertex Pharmaceuticals, along with CRISPR Therapeutics, licensed a new gene therapy, CTX001. This approach involves removing blood cells from a patient with sickle cell disease and using CRISPR-Cas9 to edit the beta-globin gene so that the cells produce high levels of fetal hemoglobin. Fetal hemoglobin is a form of protein that's usually produced only through 4 months of age, but has a high capacity for binding oxygen. When the edited cells were reintroduced into the patient's body, the sickle cell disease symptoms were significantly reduced. Patient data from the CTX001 clinical trials have shown promising results in patients. Victoria Gray was the first person to be treated for SCD using this therapy, and in July 2020, it was announced that her disease had shown significant improvement. Her fetal hemoglobin levels had shot up, and her pain bouts had vanished. More significantly, she no longer needed blood transfusions. Since then, it has been reported that several other patients with SCD have shown significant improvement as well. That's all for today. If you are further interested to know more about using CRISPR to the ethical considerations of gene modification, watch our next videos, and don't forget to subscribe so you don't miss out any opportunity.